This next section focuses on transcription. So if we think about a strand of DNA, DNA has regions on it called genes. And a gene is just a series of bases that code for a protein. What do we need proteins for? Well, proteins do everything inside of the cell. A protein is going to be able to make up the structural components of most of the cells. Proteins serve as enzymes, and those enzymes are going to be able to do metabolism, so metabolic processes. They're going to be able to create every component of the cell. So we have the instructions for making these proteins in our DNA. And those instructions will be read and turned into a protein. So the analogy that I've used before is talking about baking a cake for your aunt in Arizona. So we have a whole bunch of directions in here. We have this giant recipe book which contains all of the recipes to make anything that you would ever want to make ever. We need to find the correct recipes, so the cake recipe, but then instead of carrying this gigantic cookbook over to the kitchen counter so that we can read it and start adding the appropriate ingredients, we're going to jot down a copy of the recipe. So let's pretend that we open up our kitchen cabinet, which is kind of like the nucleus, and we start flipping through the pages of our recipe book. So we're looking through the DNA. We find the recipe for the protein that we want to make, or the cake that we want to make. Then we take a piece of scratch paper and we kind of copy down the message. We copy down the recipe. We need 2 eggs, a cup of flour, and a cup of sugar, and blah, 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 blah. So we start copying down that message. That's really what transcription is. In transcription, we're making a copy of a gene of interest. And this copy is known as messenger RNA. So, mRNA is a copy of our gene of interest. And then we can take that copy of the recipe that we wrote down, so that list of ingredients, we can take it over to the kitchen and start adding in the appropriate ingredients. In other words, our ribosomes can read what this message says and start incorporating in the correct amino acids. Amino acids have a very specific structure, and I talked a little bit about this when we went through the kind of macromolecule section of the earlier weeks of the lectures. That structure of the amino acid allows for it to fold a certain way. So we have these R groups that stick out of the tops of the amino acids, and it allows it to fold in a particular way so that we can create these 3D and structures, these functional structures of proteins. Now their exact shape is important because their shape fits a particular substrate, and we'll learn more about that when we go through the metabolism type stuff. But it's important to kind of grasp how important this is. Having a gene, these are the directions, this is the copy of the directions, and we're eventually going to turn that into a protein, which is essentially a sequence of amino acids. And the sequence of amino acids determines the folding of the protein. Which is going to determine the function. So, how do we know where this gene is located? If you do a DNA extraction and you look at the structure of DNA, it's just a bunch of goop. It's like clear goop. How does our body or our cells know where the beginning of a gene is? Well, every gene has a promoter region and a termination region. So then our next gene would also have a promoter and a termination region. These promoters can be blocked by different proteins or factors inside of the cell, or they can be available. If they're available, then the gene is going to be transcribed and eventually translated into a protein. If they're blocked, then the gene is not going to be transcribed and not turned into a protein. And we can conditionally block these and unblock these depending on the needs of the cell. So again, we're going to start talking about the process of transcription. So transcription is making an mRNA copy of a gene. And transcription has three main steps: initiation, elongation, and termination. During initiation, RNA polymerase binds to a promoter. Now this is a lot more complicated in eukaryotes. In prokaryotes, it involves the use of sigma factor, and then sigma factor kind of guides RNA polymerase to the promoter, and then as soon as RNA polymerase binds to that promoter, sigma factor falls off. When RNA polymerase binds to a promoter, it's going to separate the strands of DNA and kind of unravels them so that you can access the inside message. You can access the sequence of bases that is found in the DNA. So if we draw out the DNA here, we have our double-stranded DNA. RNA polymerase is going to open it up so that we can read the sequence of bases. And the thing about transcription and making a copy of the gene, we only need to copy one of the strands. We don't need to copy the other strand. So RNA polymerase kind of serves the role as helicase, but it also serves the role of DNA polymerase III. So not only can it open up the strand, but it can also start creating new DNA. It's also important to note that this is all bases. So we have A, C, G, T, and so forth. So when we make our messenger RNA copy, we're going to make a complementary copy. So where we have an A, since we're making RNA, it would be a U. Where we have a C, we would add a G. Where we have a G, we would add a C. And where we have a T, we would add an A. So the first segment of our messenger RNA that's being made is U, G, C, A. And it would continue down the length of the gene until it hits a termination region. Elongation is the process of adding or incorporating in the mRNA bases. Remember, U instead of T. And it's going to be complementary to the template strand. And then termination, basically all that happens is the RNA polymerase hits the termination region and this stops. There are factors that can be found inside of the termination region. Those factors include, like you have row-dependent termination. There's also row-independent termination, but basically it's just a little rock that sits in the termination region, so when the machinery kind of hits that, it dissociates. Then you have your free messenger RNA, and that messenger RNA is going to go find a protein, a ribosome. And when the messenger RNA finds the ribosome, that's what kind of starts the process of translation.